So this is an overall introduction just to the microbiology course for fall semester 2021. There are some older overviews as well from other previous semesters. Each time it's basically the same course, except some things will be different. The dates of things will obviously be different, and the way we handle the lab and some of the assignments might be a little bit different. But overall, everything is mostly the same. So if you saw some older materials, mostly the same stuff, but this is the update as well. So I'm just going to be quick with this. There's additional information. Specifically, look at the dates that are in the lab schedule and then a lecture schedule. But I'm going to go through that a little bit now. So your class is technically an online course. It's an OLCR, which means it has a campus requirement, and that requirement is the lab. So it's online lecture like this. And so the lecture videos are the main content delivery of the course. I have slides as well, and I'll talk about study guides and how to use them in order to prepare your studying and organize all the material for your exams. And I'll talk a little bit about the exams. The labs I'm going to mention up front though, because we're going to be meeting in person. However, because the lab is small and we are social distancing, you're meeting in separate groups. So you will not meet in lab every single week. You'll meet in lab every other week. And the weeks you're not in lab, there'll be worksheets, online assignments, or other things that you'll need to do at home. Some of them might be a direct connection with the lab. There might be a pre-lab or a post-lab or something just that's a different activity. But there are things you'll need to do and turn in for grade. And those will be online as worksheets. In addition, there'll be the in-person work, which is going to be largely technique-based. So you're going to be learning specific techniques and skills that are hands-on that you can't really do on the computer. And then you'll be tested on them. So there'll be technique exams, skill tests that you can actually perform that work. So let me talk about the organization of the course for the fall. And I have a quick copy of the schedule here so I could refer to dates that you have as well that you can get in Blackboard. The specific schedule is there as well. So the first thing you should do is when you go into the course, you should find the study guide for each exam. So then you go into the course content, exam folder, exam 1, for example. There'll be study guides. Essentially, it's just a list of terms that you're going to need to know for that exam. So that block of material covers a bunch of terminology. Okay, so that starts right at the very beginning of the course, which is August 6th. August 26th. Okay. And so since it's online, you're working sort of on your own in terms of timeline. So you can spend, a lot of hours, 1 or 2 days a week. You can spread out a little bit over, 4 or 5 days a week or more. So how you spread out your time is sort of up to you. On the schedule, what I have are weeks, and then I have topics and quizzes. All right, and so this is sort of your guideline for when you should do these. So quiz 1 should be done in the first week. This is because it's mostly background material that you should have had before taking the course. So if you don't know this stuff, you're going to need to learn it because we're not going to spend a lot of time on it, or we're going to go very fast through it or not cover it at all. So do the quiz, and if you have trouble with it, you'll find that all the quizzes are going to be 3 attempts. So you do a first attempt and you do really terrible on that quiz. That's fine. That's the idea, is now you're going to go back, look up the information on the things you missed or don't understand, find the answers for them, then go back and retake the quiz. It'll be questions in a different order and all, but essentially the same questions. So you can then kind of get through that material, make sure that you actually know those sort of key points. And then we get into the actual content of the course. So exam 1, so it does sort of an introduction. There's a bit on the history of microbiology, which isn't a lot of dates and things you have to memorize. It's just an overview of the progression of how certain things were discovered and with the names of a couple key people and the rough timeline of when those things happened, just to give you perspective on them as well. We then talk a little bit about microscopy and cells and cell size. So microscopes, which is also part of the lab. So there's an direct overlap with lab and the lecture here. So you will be asked some questions about microscopes and how they work and things like that on the lecture exam. And then we get into the cells themselves, which in our class are prokaryotic cells. The prokaryotic or bacterial cells. And so here we're going to look at a lot of the structure. And one of the main structures we're going to look at is the cell wall. So you're going to need to know the chemical composition of the cell wall, the arrangement of the cell wall, the differences in cell walls between gram-positive, gram-negative bacteria, alterations in the archaea, a different group of prokaryotes, and then just some general variations of the cell wall between other sort of smaller groups or subgroups of bacteria. So the core structure first, and then you kind of expand out to the diversity of that structure. And that leads us a lot into classification of bacteria, which is one of the last topics. Because classification often starts off with the type of cell wall, gram-positive bacteria, gram-negative, and you need to know what that means. What is gram-positive or negative? And that comes back to the structure. Now, when we get to the point of an exam, you will have already taken several quizzes leading up to the exam. So on this particular schedule, you will have taken quiz 1, 2, 3, and 4 before taking exam 1. The point is to do them to prepare yourself for the exam. So the exam I have in the schedule here, September 22nd, it says, to the 25th. So it's not a date, it's of a range of dates. And that's because there's going to be an online portion of the exam similar to the quizzes, but longer. And you'll have some flexibility of when you do that, but whenever you start to do it, you'll have a very limited amount of time to answer the questions. You will not be able to look up essentially the answers for all these questions, or you won't be able to finish the questions and complete them. So you need to study like a regular exam. You'll be given a reasonable amount of time to read the questions, read the answers, and then select the answer. If you know it, you just select it. If you don't and have to look it up, you'll leave many of them blank, most likely. There'll also be a written part that's in person, and we'll do that during your lab meeting. So whenever you come in for lab, it's not going to be for the same day for everybody, there'll be a part where you're going to draw sketches. So sketches of the structures of the cell wall, for example, or flagella, or the difference between flagella and pili, or other things that you'll learn as you go through these materials and lectures in the first part. So how do you know that you covered all the material? Well, the study guide. The study guide is going to again have a list of terms, and it's going to also have a list of things like sketch this, sketch that, label these. And if you can do those, that's going to be what you're going to be asked to demonstrate on your exams. Right, so very directly. And where do you get that information? Well, I have slides, so they're PowerPoint sort of slides that you can kind of go through and read. And then there's the actual lectures like this, where I'm drawing things on a board, talking and covering all the essential material in the class. Right? And you should be able to go and check everything off. And if you can't check something off, you say, I don't know where that is, I didn't see it, then you just let me know, and I can either direct you to it or help you out in person. They're also reading, so there'll be references to the textbook, which is online. There are 2 different textbooks that are online books, and you just click on the link, and it just brings you to the pages that you're supposed to be reading or get reference material from, or find the definition of terms or other pictures other than my drawings, some nicer artistic ones that you can actually see. Okay, then you're going to do the quizzes, and that will prepare you for an exam. And that's going to repeat essentially for every exam. You kind of would find a study guide, get the slides, watch all the lectures, make sure you covered all the material, take the quizzes. If you find you're missing stuff, then you go back and either find it or ask about it, and then you'll be ready for the next exam, and so on and so on. Okay, so the dates of those exams are, like I said, scattered roughly about 3 to 4 weeks apart. So you have September 22nd to the 25th, October 20th to the 23rd, November 17th to the 20th, and then the final exam. Same thing, there'll be the online and the in-person part is in December during the finals week. So 4th exam is also the final exam, which means it's 150 points. It's cumulative, so it's 100 points of the 4th exam material, and then it's going to be 50 additional points that are cumulative, so there'll be topics from exam 1, 2, and 3 on that exam. So exam 1 is essentially introduction to prokaryotic cells and the cellular structures is the main thing. There's other topics on it, and then there's like a little bit on classification, a little bit on history, and some other things, but that's kind of the core material. Exam 2, we start to get into metabolism. So you're going to go through things, some stuff you'll need from the first exam about membranes and diffusion and active transport and all. And it'll relate back to the structure, as well. But we'll go through how the cells will do processes like glycolysis and citric acid cycle and so on, which are overlap with other cell biology material. Then unique processes of fermentation and things that prokaryotic cells do. And then cell growth, so there'll be, which is actually cell division, that is unique for bacteria. The things that regulate and control that or prevent it, like antibiotics, and how they actually work specifically to stop cell division or cell growth, and what aspect do they affect the DNA, do they affect the cell wall structure, what are they actually doing? All right, so we're kind of focused really on metabolism, energetics, cell division, that kind of material in exam 2. Okay, exam 3 gets into more genetics. So DNA, gene expression, that's going to be overlap a little bit with eukaryotes, but then specifically for bacteria, what do they do different? What's different about them? What's different about the structure? What's different about some of the processes and unique things that they can do? How they regulate gene expression, and so unique processes as well, how they condense their genome, and a variety of other things related specifically to the genetics of bacteria, which will link back a little bit again to the classification, because we have some molecular component to that as well. And then exam 4, we'll go into some other groups like the archaea. We'll go into virus or virology, and some immunology. And the process for bacteria of pathogenesis. So essentially, the bacteria that cause disease, how do they cause disease? What are they doing to actually make an individual ill? What chemicals do they produce? Then how do humans or other organisms react to that? What do they do to prevent or stop that from happening? And in addition, we'll look at a virus. What is a virus? Viral life cycles, and then how does the immune system react to those as well? And that's kind of the 4th exam. So that's kind of the material of the whole semester in very, very loose kind of short detail. There's a lot more deep details that we go into with each section. So for you, what should you be doing? As the course begins, you should get your study guides for each exam, find the slides for each exam, and start watching the lectures in order. You should refer to the schedule for roughly when you should be doing those. If you see that, like, oh, geez, there's a topic here on cell wall, it says I should have covered that already by September 6th or something and haven't even looked at it yet, you're behind a little bit. The main thing is the quizzes don't have a specific date that you have to complete them by. If you don't do them before the exam, you could potentially still do them afterward, except they're not going to help you for the exam at all, and that's the point. They're designed to help you study and prepare for your exams. So each exam will have several quizzes, about 4 quizzes that you'll be able to do that will help you study and guide you toward the material on each of those exams. All right. Labs, make sure you look at there's a specific schedule for labs where there's a group A and B for labs. And you will come to lab only on your assigned week. So if you're group A, you'll be coming the first week of classes, not the week of the 26th, but the following week after that. And then the group B will follow. The group A then doesn't come that week, right? And so just if you look at the schedule, it'll see, and I sent a message specifically saying people's names, what group they're in, and then you look at the schedule and say, okay, I come these specific weeks to lab. And that's the overview of the class for the semester.